Our resource speaker for today is a university researcher at the Biotech University of the Philippines, Los Baños, where he mainly focuses in the field of plant growth, promoting rhizobacteria. He is a graduate of bachelor's degree in biology with major in genetics and a master's degree in molecular biology and biotechnology, both from the University of the Philippines, Los Baños. His research resulted in the publication of several papers and awards on the said subject. He is also a very active. He is also very active in extension activities toward promoting and teaching the use of microbial inoculants to local farmers and students across the Philippine archipelago. So, sa ating pong mga manonood, lahat po, sabay-sabay po natin i-welcome ang ating resource speaker ngayong umaga, si Sir Robert Nepomuceno. Good morning, Sir Robert.
Thank you for the kind introduction, and good morning, everyone. Today, I will be presenting the session that was originally scheduled for last week, but unfortunately, it was postponed due to the 6.3 magnitude earthquake. So the title of my talk is Sustainable Alternatives, Harnessing Microbial Inoculants for Enhanced Crop Production. I will be focusing on products that have been developed through the support of the EVAR, and these products include Oryzinc, Zinc, and Bioplasma. First, let me introduce you to the National Institute of Molecular Biology and Biotechnology, also known as the NIMBB. So NIMBB has 4 research institutes spread across the country, and each one focuses on different areas of research. First, where I'm currently affiliated with, we have Biotech UPLB, which is based at the University of the Philippines, Los Baños. Our research is related to food, agriculture, and environment. Our research is focused on developing innovative technologies and solutions that can boost agricultural productivity, improve food security, and tackle environmental challenges. Next is we have NIMBB Diliman. So these guys specialize in applying molecular biology and biotechnology to industries. So they're all about bioprocessing, bioremediation, bioenergy, and other industrial sectors. And moving on, we have NIMBB Manila. So they're all about health-related and medical applications of molecular biology and biotechnology. So they delve into areas like disease diagnostics, drug development, genetic engineering, and biomedical research. And lastly, we have NIMBB Visayas. So they specialize in research related to fisheries, marine biology, and aquatic sciences. So they're tackling challenges in sustainable fisheries management, aquaculture, marine biodiversity conservation, and ecosystem health. So let's talk about Biotech UPLB. So our institution was established on December 20, 1979. And the mandate of the institute is to develop cost-effective and environmentally friendly technologies to produce goods and services that are comparable or better alternatives to conventional products for use in the following sectors. So agriculture, environment, energy, and industry. So in simpler terms, we're all about creating affordable technologies that offer cheaper options to conventional products. So we achieve this by using locally available materials that are not only economically viable, but also environmentally safe, which promotes sustainable production practices. So in this presentation, as I've mentioned, we'll dive into 3 specific technologies that we've developed: Oryzinc, Zinc, and Bioplasma. So as I mentioned earlier, Biotech UPLB has a clear mandate in the development of alternative products for various sectors, including agriculture, environment, energy, and industry. So within Biotech UPLB, we have different programs dedicated to each sector. So for the agriculture sector, there's the Biotechnology for Agriculture and Forestry program, or the BAFP. So this program focuses on conducting research on microbial inoculants like biofertilizers, plant growth regulators, biostimulants, nanobiosensors, and animal vaccines. So why microbial inoculants? Why do we need to study microbial inoculants? Microbial inoculants offer numerous advantages in various agricultural applications, which include the increased crop yield. So this is by promoting better plant growth and development. So the microorganisms they produce growth promoting substances such as plant hormones, enzymes, vitamins, which results in healthier plants with increased vigor and resilience. Additionally, microbial inoculants, PBGB, or plant growth promoting bacteria, improves the uptake of plant nutrients. This is by establishing a symbiotic relationship with plant roots. So they create a network of beneficial interactions that enhance nutrient absorption from the soil. So another advantage of microbial inoculants is their ability to stimulate biological processes in the soil. So the beneficial microorganisms enhance soil fertility by breaking down organic matter, releasing nutrients, and improving soil structure. So this leads to improved soil health and nutrient cycling, creating a favorable environment for plant growth. Moreover, microbial inoculants act as a biocontrol agent against pests and pathogens. So the beneficial microorganisms can outcompete harmful pathogens, reducing the incidence of diseases and pest in crops. So this natural approach helps minimize the reliance on chemical pesticides, promoting sustainable and environmentally friendly agricultural practices. Also, the use of microbial inoculants reduces the application of chemical fertilizers and production costs. So, by enhancing nutrient availability and uptake, these inoculants decrease the need for excessive chemical fertilizers, saving costs for farmers while promoting a more sustainable farming practices. And lastly, microbial inoculants are eco-friendly. They contribute to restoring the soil's natural fertility, reducing soil degradation and erosion. So the most common active ingredient found in microbial inoculants are the plant growth promoting rhizobacteria or PGPR. So PGPRs offer several benefits in plant growth and development, mainly in that it facilitates nutrient mobilization and biostimulant production. So specifically, PGPRs promote seed germination rates, and enhance nutrient uptake, which leads to improved plant growth and vitality. Additionally, PGPRs exhibit phytostimulation. So, phytostimulation refers to the ability to stimulate plant growth through the production of growth promoting substances. So, they also act as biocontrol agents, helping to suppress the growth of harmful pathogens and pests. So, as can be seen in this figure, the presence of PGPRs kill off pathogens. And another advantage of PGPRs is that their ability to increase shoot and root height and weight, so which contributes to overall plant vigor. And lastly, PGPRs have been shown to delay senescence, which is just the aging and deterioration of plant tissues. This allows for a prolonged plant productivity. So our research mainly delves on this topic, and one particular topic which they were funded was our research on zinc solubilizing microorganisms. But why do we need to study zinc? So this is because soil zinc deficiency is one of the most widespread soil micronutrient deficiency in the world. So this figure provides insights on the global scale of the scarcity of zinc in the soil. So this is according to the publication of Phillips, which was published in 2015. So virtually all the major agricultural lands are zinc deficient, including the entirety of the Philippines. Most of China and a large proportion of Africa are considered to be zinc deficient. So the exceptions are the white part. So that's Russia and Canada. So that's essentially frozen tundra, and good luck farming in those areas. So another exception is the north of Africa. That's Sahara Desert. May I remind you, and also the entirety of Australia. So Australia is also practically a desert because the mountain ranges prevent rainfall from reaching the inner Australia. So that's also why most cities in Australia are situated in coastal areas. So this figure depicts the extent of soil degradation in the Philippines. Specifically, a more detailed figure highlighting soil zinc deficiency in the country as compared to the previous figure. So, as you can observe, the Luzon Plains, particularly Nueva Ecija region, falls under Group 1 and Group 3. So, Group 1 signifies deficiencies in zinc and sulfur, while Group 3 indicates deficiencies in phosphorus, nitrogen, zinc, and sulfur, which is likely caused by excessive usage of fertilizers, urea, and imbalances in nitrogen and phosphorus fertilization. So this is deeply concerning, seeing the degradation of solids in the Nueva Ecija. which is also known as the rice granary of the Philippines, as this has implications for food security in the country, given that a majority of Filipinos rely on rice as their staple food. Rice and corn crops are very sensitive and highly susceptible to zinc deficiency, so making it imperative to develop effective, sustainable, and environmentally friendly solutions to address this challenge. So in rice, symptoms of zinc deficiency include stunted growth, depressed tillering, and in the field, it shows as uneven growth of plants in one paddy and delayed maturity. So this is based on the publications, or these pictures were taken from publication of field rice, which was published in 2000. So to address this issue, we have developed two microbial inoculants specifically designed for these crops, Orizing for rice and Maisin for corn. So let's begin by discussing Orizing, its features, and its impact on rice cultivation. So Orizing is a microbial-based inoculant designed specifically for rice crops. It harnesses zinc-solubilizing bacteria, which plays a vital role in converting zinc in the soil into a readily absorbable form by plants. So by facilitating this conversion process, Orizing significantly reduces the need for excessive zinc chemical fertilizer application in lowland rice cultivation. So this ultimately addresses the crop sensitivity to zinc deficiency, and this technology offers a sustainable and cost-effective solution to enhance zinc uptake by rice plants, which promotes healthier growth, optimizing yield potential, while minimizing environmental impact associated with excessive fertilizer usage. So this inoculant, similar to Orizing, incorporates zinc-solubilizing bacteria, which play a crucial role in converting zinc present in the soil into available forms for plant absorption. So because of this, Zinc effectively reduces the need for excessive application of zinc chemical fertilizers, and this not only addresses the issue of zinc deficiency in corn, but also promotes sustainable farming practices by minimizing environmental impact associated with the overuse of chemical fertilizers. So with Zinc, farmers can enhance zinc uptake in corn plants, leading to improved growth, development, and ultimately higher crop yields while minimizing cost. Presented here is an extensive conceptual framework outlining the process of developing zinc solubilizing inoculants. So this framework encompasses various stages, starting from the isolation of microorganisms, followed by in vitro and in vivo screening to assess their effectiveness. Subsequently, the identified promising strains undergo field testing to evaluate their efficacy, along with the on-farm demonstrations to validate their performance under real-world conditions. Finally, successful strains move towards mass production and subsequent registration as commercial products. So let's first start with the isolation of microorganisms. So in this figure, you can observe a fungus exhibiting a halo zone in a medium containing insoluble zinc. So the presence of a clearance zone indicates that the isolate has the ability to solubilize zinc. So this particular isolate was obtained from cassava roots, and once we confirmed its zinc solubilizing capability, we also conduct tests to determine other characteristics, such as whether the isolate is also capable of nitrogen fixation, which is of significant importance. And in addition, we also tested for cellulose degradation capacity as well as lipolytic and amylase activities. So what comes next? So what comes next after the laboratory testing? We proceed to test the isolates on actual plants. So the figure presented shows the in vivo screening results of the top zinc solubilizing isolates on rice. So these results emphasize the potential of these isolates in effectively addressing zinc deficiency in rice crops, as the plants treated with the bacterial isolates exhibit comparable results to the positive control. So these findings and figures were published in 2020. And if you're interested, You can access the publication online as it is publicly available for further exploration. So the citation is listed there. Later on, you can request for a copy of the PowerPoint, so you don't need to copy or screenshot that now. And after conducting tests in the growth room, we proceeded to evaluate the isolates in actual field conditions. Specifically, we conducted the field trial in Victoria Laguna, which is known to have zinc deficient soil according to BWSM. So this test was carried out in marginal soil and with zinc deficiency. So this explains the below average performance of the control group. So the average yield for the country as of my last check was 4.1 tons per hectare. So the graph presented illustrates the yield of rice when inoculated with a zinc solubilizing isolate. So based on the figure, certain isolates such as A1 and D6 showcased significant improvements in comparison to the positive control, with yields reaching close to 4.5 tons per hectare in marginal soil conditions. So the on-farm demonstration of the developed zinc solubilizing inoculants has received positive feedback and commendations from the farmer cooperators across various sites. So these on-farm demonstration trials aim to showcase the practical application and effectiveness of these inoculants in real life farming conditions. So the feedback from the farmer cooperators has been highly favorable, indicating their satisfaction with the performance and results obtained from these inoculants. And the farmer cooperators have expressed appreciation for the noticeable improvements in crop growth, health, and overall yield, which was achieved through the applications of these inoculants. One notable observation we made when using orizing in rice is that it enhances the resistance of the plants to lodging. So preventing them from easily bending or falling down. So, based doon sa figure, doon sa control, kita nyo yung mga nakadapa na rice as compared to the inoculated plants na lahat ay nakatayo pa. So, this figure provides clear evidence of the impact of rising in rice. So a comparison between rising inoculated rice field and the control treatment shows a significant difference in the occurrence of lodging, which is particularly evident. This was actually Typhoon Julina, which is a signal number 2 typhoon last September 2021. So based on this observation, the application of rising contributes to the development of climate resilient rice, which is crucial for ensuring food security in our country, which is particularly important because our country is frequently ravaged by typhoons from the Pacific. During harvest time in Victoria Laguna, we observed significant differences between fields treated with zinc or RI zinc and those without any zinc application. As you will remember, the Victoria Laguna was determined and reported to have zinc deficient soil. So figure A illustrates the neighboring fields where no zinc or RI zinc was applied. This shows sparse growth and delayed maturity, which are typical indicators of zinc deficiency. So these pictures were captured simultaneously. They were planted at the same time. So this highlights the effectiveness of zinc in addressing soil zinc deficiency, resulting in early maturity and significantly higher yields compared to traditional farming practices. So our main focus was rice and corn, and this is the results for corn. The graph presented illustrates the results of a field experiment conducted on corn intercropped with cassava. So specifically focusing on the effect of inoculating seeds with maize. As you can observe, some isolates, particularly A1, has a greater yield as compared to the control. and corn plants with the recommended rate of chemical fertilizer. The data clearly shows a noteworthy increase in corn yield when the seeds were treated with maize compared to the non-inoculated plants. We also tested the inoculant on actual field conditions, particularly in the zinc deficient soil of Victoria Laguna. This figure shows you the cornfield of a farmer cooperator, Ms. Lonar. She has expressed satisfaction and contentment with the noticeable yield improvement achieved through the application of the inoculant. We also promoted the products to local farmers. So multiple farmers' field days were organized across various regions in the Philippines to conduct informative lectures on the significance of these microbial inoculants. And also, we want to educate the farmers on their proper usage and application. So during these farmers' field days, we actively engage with farmers, providing valuable insights into the benefits and practical applications of these microbial inoculants. So the lectures aim to enhance farmers' understanding of how these inoculants can contribute to improve crop productivity, disease management, and sustainable agricultural practices. And as a result of of the project, two technologies named Rising and Mazing were successfully developed and has now successfully entered the production phase. So these technologies have garnered significant interest from farmers in the vicinity, and they have incorporated them into their agricultural practices. Rising and Amazing offer a promising solutions for addressing zinc deficiency in rice and corn crops. So our team at BioTech has also developed extension materials, including brochures, with the aim of providing information to farmers and other stakeholders regarding the benefits and usage of these microbial inoculants. So these extension materials serve as an essential tool to bridge the gap between scientific knowledge and practical application through user-friendly and concise formats. And I would just like to highlight the numerous awards and recognition received by our team. We have been honored with several Best Paper and Poster awards in prestigious conferences held in the Philippines, Malaysia, Indonesia, and Japan. Notably, our paper received recognition from NSARD and various national conferences in the Philippines. Furthermore, we were honored to receive the prestigious Hitachi Asia Innovation Award, which acknowledges efforts in the ASEAN region that contribute to the UN SDGs or the United Nations Sustainable Development Goals. And we are immensely grateful to the ABAR for their funding support, as none of these achievements would have been possible without their valuable assistance. Our team has also implemented another project funded by the ABAR. This time, we focus on combating cassava phytoplasma disease. So cassava phytoplasma disease, or CPD, it is a significant threat to cassava crops, which can cause up to 100% in yield loss and also resulting in a reduced yield quality. The traditional method used in controlling CPD is through the use of antibiotics or streptomycin in the cassava cuttings before planting it. So this method is not sustainable and could potentially lead to the development of antibiotic-resistant microorganisms. So during that time, an alternative was urgently needed. So through this project, we have successfully formulated an inoculant coined bioplasma. So when it when it's combined with VAMRI, it acts as a preventative measure against the occurrence of CPD and effectively regulates the severity of the disease. So, by the way, VAMRI is a mycorrhizal inoculant, and it's just an acronym for vesicular or vascular mycorrhizal root inoculant. So this conceptual framework outlines a systematic approach in studying and addressing CPD. So this includes the identification of potential biocontrol agents, evaluation of their efficacy, and consideration of other economic aspects. So, in the process of in vitro screening for the biocontrol of CPD, various isolates derived from the soil and roots of CPD infected fields were examined. So, we are particularly interested in indole acetic acid or IAA. So, IAA is a plant hormone known for promoting root development, enhancing nutrient uptake, and stimulating overall plant growth. So IAA can potentially enhance the growth and development of cassava plants, which contributes to their resilience against CPD. We also tested the isolate for phosphorus and potassium solubilization. Isolates that exhibit phosphate or potassium solubilization can enhance the availability of these nutrients in the soil, which supports healthier plant growth and increasing resistance to CPD. Another attribute assessed is the production of ACCD amines. So, and this is an enzyme involved in the metabolism of a plant hormone called ethylene. So, high levels of ethylene can lead to stress responses in plants, including susceptibility to diseases. So, isolates that possess ACCD aminase can potentially mitigate the negative effects of ethylene and alleviate stress in cassava plants, enhancing their ability to withstand CPD infection. Next, we'll try to figure out the best microorganism for use in tandem with memory. So the results indicated that one specific bacterial isolate, CSV51, exhibited the most promising performance in terms of cassava crop yield. So compared to the negative control and other treatment groups, the cassava plant inoculated with CSV 51 showed notable improvements in growth parameters and overall biomass production. So in the next trial, the focus was on determining the survival rate of cassava plants under different treatments listed here. So the results show that the cassava plants treated with CSV or isolate CSV 51 and BAMRI exhibited a remarkable 100% survival rate. So this outcome suggests that the combination of isolate CSB51 and BAMRI has a positive effect on the survival and overall health of cassava plants. I would just like to make it clear that all planting materials mentioned in all these trials were gathered from areas with CPD, specifically those expressing Rosette syndromes. Symptoms rather. So all these measurements are under the assumption that the planting materials are infected with the pathogen. So in addition to the field trials and on-farm demonstrations, we conducted field setups of bioplasma in different locations, including Sariaya, Quezon, Magdalena, Laguna, and Isabela. So these field setups aim to assess the performance of bioplasma in diverse geographical regions and farming systems, and also to ensure the effective utilization and proper application of the developed biocontrol for CPD. A series of training seminars were conducted in various locations, including Laguna, Nueva Vizcaya, Ilocos Norte, and Nueva Ecija. These training seminars aim to build the capacity of users, including farmers, agricultural extension workers, and other stakeholders. So the technology developed, bioplasma. So it represents a significant technological advancement in biocontrol of CPD in cassava. So in order to effectively communicate the benefits and methods of using bioplasma, IEC materials such as this brochure was developed specifically for farmers and stakeholders, which this brochure serves as a practical guide, providing a step-by-step instructions and highlighting the key advantage of using this inoculant as a microbial-based biocontrol against CPD. So to further enhance the dissemination of information about bioplasma, a comprehensive YouTube video was created. So it shows in here that when bioplasma is utilized, instead of the antibiotic streptomycin, farmers can experience an additional income of up to 57,000 pesos. So let me play this video for you. So it's not that long. It's just around 5 minutes in length. Cassava, locally known as balinghoy, is considered an economically important crop owing to the vast array of its prospective use, from industrial, processed food production, livestock feed to direct consumption. It was considered merely as a subsistence crop, but its recent recognition as a cheap alternative to some crops for industrial use has steadily driven the demand for it. In fact, the area allocated to cassava production has steadily risen and is currently at 300,000 hectares. The Philippines, however, is considered one of the lowest cassava producer in Southeast Asia. The country's cassava industry faces a serious threat caused by cassava phytoplasma disease or CPD. CPD has been shown to diminish yield by up to 90%, and it has been shown to negatively affect starch content and quality. CPD is caused by the bacteria called phytoplasma. They are commonly found in cassava phloem seed tubes. They lack cell wall and they are limited by a unit membrane unlike most bacteria. The symptom of a cassava plant infected with phytoplasma include dwarfism, decreased roots and leaf proliferation, and diminished internodes at the apex of the plant, giving it the appearance of a vicious fruit, which is why it's also known as the cassava vicious fruit. CPD is threatening cassava production in numerous parts of the country. The proliferation of CPD-infected cassava was caused by mass cassava production, strong reliance on monoculture of high-yielding varieties, and a lack of surveillance of suspected infectious material and its transport to other fields or farms. The negative consequences of CPD in cassava production in terms of yield vary depending on the period in which the symptoms persist. The symptoms occur 4 to 6 months after planting or in the middle of the growing season. CPD can reduce yield by 50 to 70%. When the infection occurs within the first 3 months of planting, yield can be decreased to zero due to its early death. A previous study by Velasco et al. Reported that phytoplasma infection in cassava has caused physiological changes in the plant, including the decrease of photosynthetic pigments chlorophyll A and chlorophyll B by up to 38% and 29% respectively, the increase in sugar production in the plant's leaves and roots, while decreasing starch concentration in the roots, and the doubling of concentration of toxic chemicals, hydrocyanic acid, and hydrofumarates, making cassava with CPD dangerous for human consumption. In nature, phytoplasma are spread by clone feeders such as leafhoppers and plant hoppers. As a result, a common technique for managing this disease is to use insecticides to control the insect vectors. Another method of control being used is treating the cassava cuttings with 100 ppm streptomycin solution prior to planting it. It was found effective in lowering the mortality rate of cassava plants. This control method, however, can lead to the development of antibiotic-resistant microorganisms, which will result in a more serious problem in the future. An eco-friendly, accessible, and cheap alternative must be developed. Recently, a phenomenon known as recovery has been observed in Rosaceae plant species, as well as grapevine, suggesting the effectiveness of using endophytic bacteria as a phytoplasma biocontrol agent. This instance was observed in using the treatment of either the endophytic bacteria, Pseudomonas cupida alone, or in combination with the mycorrhizal fungus, Glomus mosaic, to effectively control Chrysanthemum mielis or phytoplasma infection in chrysanthemums. Bioplasma is a natural disease control alternative that contains an endophytic bacterium that was found to have plant growth promoting and biocontrol properties. Treating CPD-infected cassava cuttings with bioplasma showed lowered occurrence rate of the disease and was observed to reduce the disease severity. The key organism was exclusively isolated from healthy cassava plants. Based on the laboratory tests and other related literature, this endophyte was observed with properties related to mineral absorption, such as phosphate solubilization, siderophore production, and nitrogen fixation. The use of bioplasma can reduce the incidence and severity of cassava phytoplasma disease, increase the source of healthy cassava planting materials, promote plant growth, increase crop yield, and serve as a more affordable alternative to chemical fertilizers commonly used in cassava. Aside from these, bioplasma is proven to be ecologically safe.
It was observed that intercropping also effectively lowered the incidence of a number of other diseases in cassava. Intercropping the CPD-infected cassava with corn allows the proliferation of the opuscular mycorrhizal fungi present in Vamri, which improves the management of the CPD symptoms together with the bacterium found in bioplasma. The use of streptomycin, therefore, is 3 times more costly than using bioplasma. We also have to consider that bioplasma is also a plant growth promoter, thus it will provide more savings from reduced fertilizer costs. Taking everything in consideration, you would only need around 6,600 pesos in fertilizer and streptomycin costs for a 1 hectare cassava plantation. Using bioplasma in combination with the mycorrhizal inoculant such as Vamri would only require you to apply fertilizer at half rate. Thus, you would only need to invest 3,500 pesos per hectare of cassava plantation, which translates to around 3,100 pesos savings per 1 hectare. So, what is the mode of application for bioplasma? In a clean container, such as a 15-liter pail, add 2 packs of bioplasma. 2 packs of bioplasma is sufficient for 1 hectare. Pour water into the container and mix until the mixture becomes slurry. Add in 1 pack of barley per 2 packs of bioplasma. Once properly mixed, soak the cassava cuttings and make sure the ends of the cutting are thoroughly covered with the mixture. Set aside for 30 minutes and plant the cuttings with bioplasma immediately. And as was mentioned, 2 packs of inoculant together with 1 pack of bamboo can be used in 1 hectare of CPD infected cassava plantation. Just some additional reminders in using bioplasma. Avoid exposing the inoculant to direct sunlight and store it in a cold and dry place to prolong its shelf life of 6 months.
Uh, so, so moving on, the successful completion of FPA registration marks a significant milestone for Bioplasma. So this registration ensures that the production and distribution of this inoculant are officially licensed and it complies with the regulatory standards. It provides assurance to farmers and stakeholders regarding the quality, safety, and efficacy of the product. So, with these developed inoculants and research and extension efforts, the accomplishments achieved thus far have created a strong foundation for further expansion, sustainability, and advocacy of the biofertilizer program, ensuring its continued success and contribution to agricultural development in the Philippines. Specifically, through expanding project reach geographical coverage, the research generated much interest that clamor among other regions for inclusion in the project is increasing. Second is that we focused on maintaining sustainability of the project through linkage with LGUs and SEUs. Another aspect of maintaining the sustainability of the project is through trainers training to strengthen the technical support needed for a nationwide adoption of the various technologies. And finally, we have to emphasize that there is a clear need to strengthen the advocacy effort for increased government support to the biofertilizer program. So, so this is related to the previous statement that there is an urgent need for the scaling of the production of the technologies for nationwide utilization for organic agriculture, food security, and sustainable agriculture. So, in conclusion, the urgent need for upscaling of this technology production stems from the critical role it plays in promoting sustainable agriculture. So, by expanding production, uh, ensuring widespread availability, we can empower farmers with effective and eco friendly solutions in achieving agricultural sustainability and addressing pressing challenges in the realm of food security, food production. Thank you very much for listening. If you're interested in collaborative research or want to try out our products on your farms, or if you want to do your thesis here in biotech, UPLB, or need anything specific, you can contact us using the following details. Also, please feel free to request a copy of this presentation for your own records. Thank you very much. That concludes my presentation.
Hello, Chuck. Thank you so much, Sir Robert, for the very informative and of course, siguradong kapaki-pakinabang po na maliban sa impormasyon ay yung produkto po ano or teknolohiya na atin pong nalikha sa pamamagitan ng pananaliksik. So bago po tayo mag-proceed sa ating Q&A, pwede po bang sa ating mga live stream viewers, pwede pong pakihit yung ating heart reaction para naman pasalamatan si Sir Robert? Pwede rin po tayo mag-drop ng comment and questions as well dito sa ating live stream. Ayan. And we also would like to acknowledge yung mga viewers po natin. Ano? Sabi po ni Sir Frank Juan, very informative daw po yung inyong tinalakay, Sir Robert. Maraming salamat. And that's the same, ayan, that's the same commendation po from Sir Vicente. Very interesting topic. So ngayon, Sir, mag-proceed na po tayo sa ating Q&A. Bilang katunayan po, napakarami po natin tanong. So definitely, yung ating pong mga viewers na halo-halo. Meron po tayo dyang farmers, meron tayong researchers, posible meron din tayong mga students na nagtitake ng agriculture-related na courses. So ayan po, kaya medyo iba-iba yung ating, yung pinanggagalingan ng tanong. To start off po, may tanong po tayo dito. So kanina po tinalakay natin, sir, yung mga magagandang epekto no, netong mga products natin. Basically, technologies na nagresulta from researchers. Ano po kaya yung mga posibleng hindi magagandang epekto, sir? Especially kapag na-overuse yung products natin like the Orizing and the Mizing.
Actually, wala naman po siyang negative effects kasi ano po ito, based ito sa living microorganisms. So kung sobrang yung i-apply nyo, kung kulang yung resources nila, hindi sila magpa-proliferate, mamatay din sila eventually. Lalo na pag walang nakatanim. So, to answer po, wala pong negative effects.
Ayan, which is good. Ano? Parang siguro yung viewers po natin, they have this idea na katulad ng gamot, ano? So when you're treating a disease, for example, pag na-overdose ka naman, merong negative effect. So good thing about these technologies ay dahil nga sila ay basically microorganisms ay hindi naman nakakasama ano if in case na overuse. But of course, we would like to advocate the right use definitely of these products. Another question po, sir, ay ayan, may mga interesado po, sir, na mga viewers natin. Available na daw po ba sa mga stores or saan exactly makakabili nitong ating pong produkto, yung Mizing, Orizing, tsaka yung Bioplasma?
Yung pinakita ko pong kanina, email address sa biotech.uplb at up.edu.ph. Pwede po silang mag-email doon. At pwede rin po namin ipadala yung mga inoculants kahit nasa malayo po sila. So, meron pa kaming last order, nasa Isabela sila. Pinadala lang din po namin yung orders nila for 500 packets.
And sir, for the benefit of our viewers po, pwede po bang pakirepeat natin, sir, yung ating email ad na pwede pong kontakin ng ating mga viewers if in case they would like to purchase the products?
Ito po. biotech.uplb at up.edu.ph.
Ayan, thank you, sir. So another question po ay a while ago, tinalakay po natin, although the name of the product itself suggests na yun yung mga crops na paggagamitan. So kanina po meron po tayong nabanggit po natin, sir, na ang Pilipinas ay kasama unfortunately sa mga lugar na zinc deficient by nature. That's why we have these interventions, ano, sir. So maliban, sir, sa palay at saka sa mais, ano pa ba sa mga typical na itinatanim ng ating mga magsasaka, yung lubos na tinatamaan or naaapektuhan ng zinc deficiency?
Usually po, ang tinatamaan talaga nitong zinc deficiency, yung mga cereal crops. So, una nga po yung corn. So, meron pang ibang other cereal crops like sorghum, wheat po siguro. Pero ang focus lang po kasi namin and registered lang po itong product ay sa corn at saka sa rice. So, hindi po namin siya pwedeng i-promote outside doon sa registration namin sa FPA.
Ayan, thank you, sir, for the clarification. So, maliban sa corn and the rice, ano, marami pa rin palang mga crops ang naapektuhan o tinatamaan ng zinc deficiency. And a while ago, you've made mention of yung manifestations like hindi lumalaki ng tama, definitely yung bunga hindi ganun kaganda. Pero ayun nga, ito pong produkto natin paglilinaw lamang ay suitable lang and registered lang po dun sa mga crops or commodities na nabanggit ni Sir kanina, which are specifically the rice and the corn. Ngayon, sir,
Gumagamit din po sa ibang crops, pero hindi po namin ma-assure yung effectivity ng product doon. So, lalo na po sa ginagamit din nila yung, for example, Bamri sa coconuts, pero registered lang po ito sa corn, parang ganun po.
ayun, actually, yun nga rin po yung in connection to that, sir. Since hindi naman natin siya pa mapopromote na gamitin sa ibang mga crops, eh, may mga crops din pala na tinatamaan ng sick deficiency or may mga manifestations. Ano kaya, although this is definitely outside of the presentation, pero in general po ba, sir, given na may zinc deficiency nga sa Pilipinas at maraming crops ang tinatamaan, ano kaya yung mga practices na madadali na pwedeng for the meantime, hanggat wala pang produkto specifically for those crops, ay pwedeng gawin ng mga farmers natin to manage the effects of zinc deficiency?
Isa po ang kailangang kontrolin natin is yung overusage ng urea. Imbalances po sa nitrogen and phosphorus fertilizer, nakaka-affect din po yan sa availability ng mga micronutrients sa soil. So isa po yun sa mga main na kailangan nating take into consideration. So ngayon naman po, meron naman po tayong zinc products, zinc sulfate, zinc oxide na pwede niyo apply sa halaman, lalo na doon sa mga areas na zinc deficient. Ang problema nga lang po kapag itong soil natin ay degraded or masyadong mataas yung pH, itong mga fertilizer na zinc ay nako-convert lang din, nako-convert into forms na unavailable for plant use. So, eventually, mag-apply ka, mako-convert sa other forms ng zinc na hindi absorbable, hindi na-absorb ng plants. So, kailangan mo ulit mag-apply later on. So, ito yung epekto talaga ng soil degradation. So, nalilimit yung availability ng nutrients, micronutrients sa soil, depende dun sa condition ng lupa.
Ayan, thank you, sir. Thank you, Sir Robert. So sa ating pong mga practitioners, farmer practitioners, it is very important na we know these technologies, na hindi lang basta-basta nag-a-apply ng fertilizer. We cannot just overuse, especially yung mga chemical fertilizers and pesticides natin kasi hindi natin alam, baka hindi na pala nakakaganda or hindi na pala beneficial yung effect niya sa ating mga pananim.
So yung
That's why it
overall,
is, yes, sir.
yung over-application ng fertilizer, hindi siya talaga sustainable for long-term farming. Eventually, madedegrade yung quality ng soil natin at pag na-degrade yung quality ng soil natin, bababa yung yield. So, ang mangyayari, mag-apply lang tayo na mag-apply ng fertilizer para mag-compensate dun sa yield loss. So, kailangan natin itong i-take in consideration.
Thank you, sir. Another question that we have is from Miss Karen. So she is asking, sir, what is the mechanism of action of the endophytic bacterial agent present in bioplasma on phytoplasma causing the CPD? And a follow-up question, do you think this product will work with the different phytoplasma strains associated with CPD? I suppose this is ZPD.
So, hindi po namin na-try sa ibang strains. So, hindi rin po namin siya ma-endorse for other strains ng phytoplasma sa ibang lugar. So, ginamit lang po namin ito for CPD. So, dun lang po namin nakita na effective itong product. So, as for mechanisms, wala talagang clear mechanism for this particular endophytic bacteria. Pero may mga publications na nagsasabi na itong mga endophytic bacteria na ito ay nag-i-induce ng increased systemic resistance doon sa halaman para malabanan niya yung invasion or infection nitong pathogen. Ay, si Dr. Alviar pala to. Hi, ma'am.
Ayan, noted, sir. Thank you. Kaya nga po ang ganda ng ating nagiging platform or avenue din itong ating seminar ano para ma-communicate din natin yung mga magagandang resulta ng mga research na nagawa ng mga expert na katulad po ninyo sa ibang researchers ano. Ayan, another question po is although this is connected to what I have asked a while ago. Although hindi pa po ninyo napopromote sa ibang crops yung product, this is from Miss Jessica again. Meron na po bang ongoing studies for the usage specifically sa ibang vegetables and CPD probably vulnerable
Yes po.
commodities?
Meron po kaming farmer cooperator sa Nueva Ecija. Ginagamit po niya itong Orizin sa maraming vegetable. Ginagamit niya sa, aside from rice, ginagamit siya sa tomatoes, sa sitaw. Meron po siyang isang malaking parang closed area for vegetables. Ano pa nga ba? Sa sitaw at tomatoes lang po yung natatandaan ko na ginamit niya. Sabi naman po niya ay effective doon kasi pinakita po niya yung difference doon sa tomatoes na walang orizing at saka meron. Kitang-kita po na mas maraming bunga yung treated with or inoculated with these products.
Okay. Would you like to add up something, sir? Or should I proceed to the next question po?
Sige, move on na lang po tayo. Pero, pwede po bang i-ano ko later on? Mag-add ako? Kasi hindi ko pa maalala ang top of my head kung ano yung ang mga vegetables na ginamitan nitong orizing. Yun nga po, tomato at saka sitaw lang po yung naalala ko. Pwede ko po bang email na lang si ma'am kung sino po yung
Yes, sir.
magtanong?
Opo.
Para
All our
mas
viewers, pwede din po
later on.
talagang mag-coordinate or magtanong sa ating mga resource speaker. We can also coordinate for them on their behalf po. So,
Sige po.
let me just proceed to the next question, sir. Meron po bang particular na kondisyon sa kapaligiran or pwedeng type ng lupa kung saan pinaka-epektibo daw po yung microbial inoculants ng nasa ating mga produkto, sir?
Effective po itong mga microbial inoculants sa mga degraded soil talaga. Doon po sa depleted na yung nutrients, doon po sa may mataas na pH na yung soil, na hindi na available yung mga micronutrients natin kasi na sila, na-convert na sila into unavailable forms na hindi na sila ma-absorb ng halaman. So, doon po. Pero kung healthy naman po yung halaman natin at available naman po yung zinc, parang hindi po siya ganun ka-effective. Or yun ba yung term doon? So, mas effective po siya sa mga depleted at highly degraded soil.
And so dun po sa mga interesado kasi we have a lot of questions po concerning the possible purchase of these products. So maganda rin pong malaman natin na this works best definitely to degraded soil, mga depleted yung mga nutrients and microorganisms. Sir, in connection to that, so since marami po tayong mga viewers na very much interested dito sa ating produkto and technology at the same time. Meron daw po bang mga ongoing capacity building on how these products should be applied or maybe how important these microbial inoculants are at paano daw po sila magiging involved sa mga ganitong seminar, training or capacity building, sir?
Actually, madami po. Ngayon po actually, meron kaming seminar training pero since magpapresent ako dito, hindi po ako nakasama. So, nagpunta po silang Nueva Vizcaya para i-promote itong mga microbial inoculants na ito doon sa area. Ituro kung paano gamitin. So, nag-invite po sila ng farmers, mga agricultural extension workers para maituro kung paano gamitin. Nag-distribute din po sila ng samples ng mga microbial inoculants natin. Hindi ko lang po sure kung anong next na sked namin. Puwede ko po bang, makuha ulit yung email nila para ipakita sa kanila kung saan kami naka-schedule na pumunta para
Yes, sir. Or yes po, sir. Or maybe
mag-training
they
nila.
can also communicate through the email that you have posted a while ago, and
Opo,
maybe they can just send their queries there, ano, para hindi naman po kayo mahirapan sa dami na magtatanong sa inyo.
kung hindi
So
po
saan
naman
po
po
naman,
sila mapupuntahan, puwede naman kami mag-send ng, brochure, saka sample nung, products. Kasi meron
ayan.
naman po instructions do'n kung ano, paano gamitin, saan ise-store, et cetera, et cetera.
Ayan, okay. So sa atin po mga viewers, ano, you can check the comment section po. Nandiyan po yung email address kung saan pwede po tayong mag-coordinate or magtanong sa kay Sir Robert and his team as well. Sir, we have last two questions. I hope you are still okay with these. Second to the last question, how long does it take to show the effect of using these products, namely the Orising and Mazing? At gaano po katagal bago magkaroon ng reaction ang Orising at Mazing sa soil?
Ito po kasing Orising, ang epekto talaga ay sa halaman. So, kung, limited yung nutrients, makikita mo na agad yung resulta nitong application nitong mga inoculants na ito during development. Kasi 'di ba pinakita natin kanina itong rice, kapag, zinc deficient, mabagal siyang, zinc deficient yung soil, mabagal mature, tapos yellow-green, tapos mababa yung height. So, doon pa lang makikita mo na kapag mabilis siyang, lumaki, ang bilis siyang mag-mature at, healthy yung ano niya, yung growth niya, makikita mo na effective itong mga inoculant na ito. Pinakita rin namin yun doon sa harvesting kanina, kasi dun sa isang side ng, field, nag-apply ng Orising at saka ng, ganitong mga inoculants, kitang-kita na yellowng-yellow na, mature na, ready na for harvesting itong, nalagyan natin ng produkto. Sa kabila naman, sparse growth, may maliliit na tubo doon sa hindi nilagyan ng Orising, tapos green pa siya. So, made-delay yung, harvesting doon sa kanya.
Ayan, thank you sir. So basically po sa ating mga farmer practitioners ay yung qualities ano makikita agad natin. Kasi nga
Opo.
yun dahil sa intervention or sa tulong ng Orising or Mazing or even bioplasma na ginagamit or gagamitin po natin. Ayan, so I guess we're all good na po sa ating mga tanong. Sir Robert, maraming salamat. Inom ka po muna ng tubig. Maraming salamat po for the information and sharing your time and of course expertise at ganin din po kay Dr. Brown na unfortunately hindi po natin kasama because of an equally important event. So regards po kay Dr. Brown at sa buong research team po and the entire institution as well din po. Maraming salamat po and more powers.
Thank you din po for having us.
And sir, we would like to award this certificate of appreciation po. And so let me just read the citation po and soon we'll be handing the certificate to you, sir. So for the meantime, allow me to read the citation. So the Department of Agriculture, Bureau of Agricultural Research awards the Certificate of Appreciation to Sir Robert A. Nepomuceno for sharing his invaluable knowledge and skills during the seminar titled Biofertilizer, Biostimulant, and Microbial Pesticide for Sustainable Agriculture, Resilience, and Awareness, held today, 22nd of June 2023 via StreamYard and DA Bar Facebook page. Maraming salamat po, sir.
Salamat din po.
Sa ating po mga viewers, baka naman po pwedeng pakihit ulit yung ating mga heart reaction or maybe the thumbs up. Pwede niyo po yata i-customize yan to show our appreciation to Sir Robert.
Tapos kung may naisip din po kayong questions, mag-email lang po kayo doon sa provided na email. Sagutin po namin.
Ayan, and before we end our seminar po, just a reminder sa ating pong mga viewers, paki-fill outan po yung attendance form. That's for the FB Live viewers at sa ibang participants po natin, those who are here sa StreamYard, yung ating evaluation form. And so we can use that to continually improve our seminar. So once again, maraming salamat Sir Robert Nepomuceno. More powers at sa lahat po ng ating mga researchers, pagbati din po Sa ating pong mga magsasaka at mangingisda at lahat ng kabalikat sa patuloy na pagpapaunlad sa sektor ng agrikultura, tuloy-tuloy po nating subaybayan yung ating monthly seminar. Kasi nga po, yun, every month ay iba-ibang technologies, iba't ibang interventions, innovations, produkto yung ating pong itinatampok. All for the same purpose, na matulungan po natin yung ating mga magsasaka at mangingisda. Maraming salamat po. This has been Jello, your seminar's host. Maraming salamat. Wishing you all a good day and God bless. Thank you, Sir Robert.
Thank you din po.